Welcome to the Chemistry, Biology and Math Revision Hub. Today we will be going through the Pearson Edexcel International A-Level Biology Unit 6, the sample assessment paper. This paper was 50 marks. I will go straight to the first question. The first question says, the photograph shows a germinating pea seed. So it says, the embryo in a seed will start to grow when the temperature, moisture, and conditions are favorable. This is growth, which is basically germination. Favorable conditions will include the right temperature, or we need the moisture, which is basically water, as well as availability of oxygen. These are very important during germination or growth of the embryo, basically. So they said the seed contains food supply. The example here could be starch, which is broken down by the enzyme. An example of an enzyme is amylase, and this provides substrates for respiration. Now, if a substrate is provided for respiration, it means we might have something like glucose, and then this combines with oxygen, producing carbon dioxide and water. So in this experiment, how can we measure the rate of respiration? It means because oxygen is produced, oxygen is used up. The carbon dioxide produced can be eliminated from of course, the respirometer by using soda lime. The soda lime will absorb the carbon dioxide, and then any gas left over is going to be oxygen. So we will monitor the oxygen used up within a specific time, and we can use that to calculate the rate. So I put here an example of an equipment which is a respirometer that can be used, and I'm going to use this to explain the explanation, the description in the next part of this question. So assume these are not organisms, but we have seeds here. The seeds are going to produce carbon dioxide that is going to be taken away by the soda lime. However, the colored ink is going to move as the oxygen is used up. In here, we have only oxygen. In that whole tube, we have oxygen. And then here we have oxygen, and inside here we have oxygen. This part is for refilling. If we want to refill, this tab can be opened, and then we refill. The color solution will return to its original side, level, or original position, and then we can be able to know how much oxygen we have used to refill in order to look at the volume before and after in the syringe and predict how much oxygen has been used up. So let's go to the next part of the question. They say, describe an experiment to investigate the effect of temperature on the rate of respiration of germinating pea seeds. So since we are studying the effect of temperature, it means change in temperature is going to be our independent variable. And then we are observing the rate of respiration. It means the dependent variable is going to be measuring the volume of oxygen used up in a specific period of time. So here we begin. The dependent variable is the volume of oxygen used up. Again, this is respiration, so it uses up oxygen. We will set up 5 different experiments at 5 different temperatures. I chose 0, 10, 20, 30, and 40. And then we will look at the respirometer and observe the distance moved by the colored liquid. As the oxygen is being used up, the colored liquid is going to move forward in the respirometer, and then we will use this formula. Volume is pi r squared L. Sometimes they say L in the XML H to find the volume of oxygen within the tube. You could use this, or you could use the refill method I've already described. If you use this method here, the L will be the length of the distance moved by the ink from the original position to the position when the experiment is stopped. And then the experiment is carried out at a specific time. Example could be 10 minutes, it could be 30 minutes, depending on you. And then we have to make sure that temperature does not change during this experiment. So we'll control the temperature using a thermostatically controlled water bath, and then we'll control pH using a buffer. And then, of course, we have to repeat the experiment and calculate the mean in order for our results to be valid. Part B says, state 2 variables other than the independent variable that could affect this experiment. So the independent variable is basically, we saw the temperature change, or we're changing the temperature. So the age of the seed, the species of the seed, the number of the seeds used per experiment, and the availability of water could all affect the results. Next part. They say, name one of the variables you have identified in B. I chose the age of the seeds. And they say, state how this variable could be controlled. We could control this by using seeds from the same pod. That way I can make sure at least there is going to be closeness in age if they are from the same pod or at least from the same plant, and then suggest what effect not controlling this variable could have on the results. The results will not be valid because if the seeds are older and some are younger, older seeds and younger seeds are not going to have the same rate of respiration. So maybe older ones could have a higher respiration rate than the young ones, which could confuse the result interpretation. So lastly, from part 1, it says, explain how a lack of oxygen could affect the growth of seedlings after germination. Now we know after germination, the seed is going to be able, of course, to continuously carry out respiration. So oxygen is required in respiration to produce ATP. And this ATP is useful in running cellular processes like protein synthesis. Let's say protein synthesis. So here proteins are going to be made, and the proteins are responsible for growth and structure within a plant. So if there is no oxygen, there will be no respiration or less respiration, or they could be anaerobic respiration. And anaerobic respiration produces less ATP. So meaning the protein synthesis will not occur as required, and therefore the structure of the plant is not going to be made, meaning there will be less growth or no growth. So lack of oxygen will reduce growth, and then because oxygen is required for aerobic respiration to provide ATP, lack of oxygen will lead to anaerobic respiration, generating less ATP, and then less energy would be available for cellular metabolism and growth. This brings us to the end of question 1. Let's continue on to question 2. Question 2, the photograph shows Daphnia, a freshwater animal. This is the Daphnia we're looking at. Daphnia is transparent and the heart can be seen beating. So we can see the heart of this beat. So we're going to see a student investigated the effect of caffeine concentration on the heart rate of Daphnia. If we are investigating caffeine concentration, so it means we are going to change the concentration of caffeine. So this one here is going to be the independent variable. Independent variable. And then the heart rate of Daphnia is going to be basically, we're going to see how the heart rate is going to be changed. The student selected 5 Daphnia, so there is A, B, C, D, and E. The student measured the heart rates in beats per minute of each Daphnia plant. This was repeated using 5 different concentrations of caffeine solution. So you can see this is the independent variable. We change the concentrations of caffeine and then observe the results. Let's go to the next part. So here they say the table shows the data from this investigation. Here we see for each Daphnia, of course, that was used in this experiment, and they have the mean heart rate that was calculated. So the first question says calculate the mean heart rate for 5% caffeine concentration. At 5% caffeine concentration, to find the mean heart rate, we have to add everything, that to that to that to that to that, and divide everything by 5 because there are 5 different samples, and then the answer we get is 2.81 beats per minute. That is the units for this. So we go to the next part. It says plot a suitable graph to show the effects of caffeine concentration. Now, effect of caffeine concentration, this means it's going to be the independent variable, therefore, on the x-axis. On the mean heart rate, it means the mean heart rate is going to be on the y-axis because it's the result, therefore, it's the dependent variable. The heart rate was measured, and then this was the independent because it is the one that was changed. So I'm going to zoom and show you like that so that you can see the whole thing. So you can see here we have the caffeine concentration, again percentage on the x-axis, and then mean heart rate in beats per minute on the y-axis, and the data was plotted as you can see. All we needed to do here is just plot the points. You do not have to connect them, and then you get the 3 marks provided. You show the labels as well as the units and the exact points. You would get all the marks required. So I will go to the next part. The next part says, state a suitable null hypothesis for this investigation. Remember this investigation, we are trying to see if the mean heart rate is actually increased when the caffeine concentration increases. So the null hypothesis should be there is no significant correlation between the caffeine concentration and the mean heart rate of Daphnia. No correlation. And the next part says explain one ethical reason why the student chose to use Daphnia for this investigation. This answer is going to be similar every time. If we use organisms in an investigation, it means they have weak, or they have actually, the nervous system is going to be simple nervous systems, and therefore they are less likely to experience pain during the investigation. This is a very good ethical reason why some of these organisms are used in research, in biological research. The next part says the student used a statistical test to investigate the significance of a correlation between the mean heart rate and caffeine concentration. And to calculate the correlation coefficient, the student produced the following table. This is the table that was produced, and then we have the results. So they say, calculate the correlation coefficient, RS, using the formula, this one here. If we are to use this formula, we know we need to find N. We need to find, of course, the D squared, and then add everything up. So I will begin by putting, maybe connecting a table here and find the D squared for each. You're not required to do this, but I'm just doing this so that you guys can see how it's done. So I will do 0 squared becomes 0, negative 1 squared becomes 1, 1 squared is 1, 0 squared is 0, 0 squared is 0, 0 squared is 0, and then add up everything. So summation of D squared is actually equal to 2. Since I have that, I also have to find N. Remember, we have 6 different samples. So 1, 2, 3, 4, 5, 6. Therefore, N is equal to 6. So then I can substitute all those into this equation. And it will be 1 minus 6 times 2 into 6 squared minus 1, which gives me this as the answer. And I wrote it here, as you can see. This one here is our calculated value. The calculated value of RS. So we're going to find, they'll give us a table of results, and maybe we can find the critical value. So we go on, they say the table shows some critical values for this statistical test. Okay, now remember, N was equal to 6, and in biology we find our critical values as P is equal to 0.05. So I will come at P is equal to 0.05 when N is equal to 6, and this is going to be my critical value. So the critical value is 0.886. The next question says explain how the student could use the graph and the statistical test to draw conclusions from this investigation. Here we have to look at the critical values. Of course, the critical value we have got, as well as the calculated value, and then we make a conclusion. So I say, as the caffeine concentration increases, the heart rate increases. This is from the graph. From the graph, that is the evidence we saw. And also we saw that the critical value, which we have got from the table of critical results, is lower than the calculated value. If this critical value is lower than the calculated value, we reject the null hypothesis. Our null hypothesis was that there is no significant correlation between the caffeine concentration and the heart rate. So it means if we reject it, it means there is correlation, or there is a significant correlation between caffeine concentration and heart rate. And this correlation is positive based on the graph. Positive correlation here, I will say based on the graph and the data. So therefore, the correlation is positive, which exists between the caffeine concentration and the heart rate. This is the conclusion we can make. And this brings us to the end of question 2. Let's move on to question 3. Question 3, germinating cereal grains such as barley produce the enzyme amylase. The production of amylase is affected by gibberellin, a plant growth regulator. A student formed the following hypothesis. So this is a growth regulator. The higher the concentration of gibberellin, the greater the production of amylase by the germinating cereal grains. Plan an investigation to test this hypothesis. A, state 2 safety issues you would need to take into account. Now, because here we are working with plant material, and then there is a chemical involved. The plant material could have some fluids coming out of the plants that could cause allergies. And then the chemical could also cause allergies or allergic reactions to our body, so we need to use gloves. Because of this, use gloves. That is a good way to protect yourself. Also, using we have to make sure that the plant, of course, the microorganisms. Sometimes when seeds have microorganisms onto their surface, of course, you may not be able to see it, but there are some microorganisms present in there. When we allow seeds time to begin to grow, of course, the glucose is going to be provided as starch is broken down. Those microorganisms on the surface of the seeds are going to have the opportunity to grow, and when they grow, they could be able to increase in number, and if they are pathogenic, it means they can be disease-causing to humans, and therefore not good. So you have to watch out for growing microorganisms like bacteria as well as fungi. Another thing you have to find to make sure you prevent the risk of sharp parts of the plant from hurting you, as well as the sharp instruments that you might use from hurting your body. So I will go to the next part. They say describe preliminary practical work that you might undertake to ensure your proposed method would provide quantitative results. If we are doing the preliminary practical work, this means we are trying to find the optimum conditions. Okay, optimum conditions for the experiment other than the independent variable. Actually, even for the independent variable, you have to find the right concentration of whatever growth factor you're going to use. So we have to find the range of concentration of the growth regulator. Are you going to use 1.0 and then 1.5, 2.0, 2.5, and so on? Or you're going to use a different range of concentrations? You have to determine this during the preliminary practical work. And then we have to make sure, since the growth regulator is going to cause the seed to release amylase, we have to find a suitable way to measure the amylase that is released. And then we have to know how long are we going to carry out this experiment. Is it for a week, for a day, for an hour? We need to establish the time frame to carry out this experiment. And then lastly, we have to find other variables that we have to keep constant, and how are we going to maintain them constant. An example, in this experiment, enzymes are involved, so we have to make sure the temperature and the pH are optimum. They have to be optimum for this type of reaction, or for the enzyme activity to be able to work well. If they're not optimum, then these are going to be leading to a decrease in the release of amylase, which would actually be bad as well. So let's go to the next part. They say, design a detailed method, including an explanation on how you would control and monitor the variables. Now, since here we are working with a biological material, we have to always begin by stating that carry out all the experiment under aseptic conditions. This prevents contamination from occurring, so that there is no contamination. And then you state the dependent variable to the examiner. So the dependent variable is the amount of amylase produced. This is the thing you measure. So we measure the amount of amylase produced. And how are we going to measure the amount of amylase produced? We could use a Petri dish. And then in this Petri dish, we could put a starch agar. So there is starch and agar mixed together. And then we could soak the seeds into the growth regulator, ensuring that it's soaked up properly, and then allow the seeds to produce amylase. Put these seeds on the surface of this agar as amylase is being produced. The amylase produced is going to digest part of the agar, the starch agar complex. Of course, it's going to digest the agar, the starch within the agar, and then it will leave something like this. So basically, we're going to leave a clear zone around the seed. In this clear zone, we're going to have agar left, but the starch is going to be gone because it has been digested. And we can measure the diameter of this clear zone around the seed, and that can be a way of interpreting how much amylase was produced. So I say, so we'll measure the diameter of the clear zone of the endosperm, or you could use starch solution. This depends on you. So how can we do this? We set up 5 different experiments with different concentrations of growth regulators. So some seeds are going to be put in maybe a lower concentration of growth regulator, while others are going to be put in increasing concentration of growth regulator. And then we'll determine which concentration of the growth regulator is going to cause the seeds to release the highest concentration of amylase in order to digest the starch present in the starch agar solid, basically. So here we have to apply the growth regulator to the seeds by placing the seeds in a solution of growth regulator for a specific period of time. Of course, for consistency, if you choose maybe time of maybe 3 hours, soak all the seeds for 3 hours in each of the different concentrations. They have to be soaked for the same period of time to maintain consistency. And then we can incubate the seeds at the same temperature for the same time to keep the temperature constant. And then we will directly, if possible, you can directly, this part here, you could directly apply the growth regulator to the endosperm if you can, if you did not soak them into the liquid. And then you could use starch solution. This part here, again, is similar. You can either use the Petri dish one, or you could use a starch solution. If you use a starch solution, it means the concentration of starch within that solution is going to decrease, as most of it is going to be broken down into glucose. So it means you will get that solution, and before you have to know the concentration of the starch solution, and then after the experiment, you will find the concentration of the starch solution and get the difference that can be used. The difference in the concentration can be used to find the amount of starch that was there, or that was used during the experiment, or that was broken down into glucose during the experiment. This part could work, or you could use the solid agar method to be able to estimate the amount of amylase that was produced. So the next part, we can repeat the experiment at each concentration, of course, and calculate the mean. And control variables, like these are the biotic ones. Biotic variables like the type of grain, the species, as well as the age of the grains, have to be controlled. To do this, you could ensure that the seeds come from the same pod in order to minimize variations. And then the next part, we can control the variables like amount of water. Now, these ones are the abiotic variables like amount of water, the temperature. We can control temperature using a thermostatically controlled water bath, and pH can be controlled using a buffer. So that is how you can answer this kind of question. And like I said, in this method, I put two possible alternatives. For the starch solution, it's okay. There you have to do the concentration. However, if you want, you can use the solid agar starch mixture, and then here you will measure the diameter of the zone that is clear around the seed. Next part says, describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. We record the results in the table with headings and units, and then we calculate the mean. Then here we will draw the data, use the data, mean data, to draw a scatter diagram, and then we can use an appropriate statistical test to establish significance. This question is always answered the same way and is always present in any paper. Lastly, suggest 3 limitations of your proposed method. Our proposed method could have limitations in case because since we are using seeds, different seeds are going to have different concentrations of amylase, and therefore it can be released differently due to the different concentrations present. Or even different seeds could have different concentration of starch, and as this amylase is being released, it could actually break up some of the starch inside the seed before it comes onto the agar, the starch agar you have put it onto. So there is a difficulty in controlling the variables like the amount of starch available. That is very difficult. And then we can also, there are like growth regulator factors. Sometimes, or we assume in biology, sometimes it's not always one factor that is going to cause a biological process to occur. What if it's not only this one growth regulator that causes growth to occur? What if there are many growth regulators that work together to cause the growth to occur, and then you use only one, and that can limit the effects of growth or the growth regulator? And then sometimes germination could be, like I said, this part here, germination could be controlled by more than one growth regulator. That is one point. And then here, the limits are, another factor that you maybe missed out could limit the effects of the growth regulator. And finally, we can talk about possibility of contamination by bacteria and fungi. Of course, on the seeds, like I said previously, there are going to be some bacteria and fungi. They will not be active because there is no food for them to grow. However, when the seed is broken, water is available, or the moisture is available, the right temperature is available, there is oxygen, and the enzymes are released to break down starch into glucose. You will see that some of these microorganisms are going to have the opportunity to grow, and when they do grow, they are, of course, contaminating the experiment, and this will not be good, or it will not be helpful in you getting the results you desire. So it will limit the method we have proposed. This brings us to the end of this paper. Thank you for being with us, and please continue and do subscribe to our channel. I'll see you in the next video. Thank you.